So in this video, we're going to be talking about the citric acid cycle. We've already talked about the glycolysis reaction in which we took 6-carbon glucose and converted it into 2 molecules of pyruvate. In this reaction, we made 2 NADHs and 2 ATPs. In pyruvate processing, which in eukaryotic cells occurred in the mitochondrial matrix, we took 2 molecules of pyruvate and converted it to 2 molecules of acetyl-CoA. And the enzyme that did this was pyruvate dehydrogenase. In this reaction, we made 2 molecules of NADH, 1 per turn of the reaction, and we released 2 molecules of CO2. Okay, so now we have 2 carbons, molecule of acetyl-CoA, and that's what's going to enter the citric acid cycle. So in the citric acid cycle, what's going to come out are eventually 4 molecules of CO2. So for every acetyl-CoA that goes in, those carbons will come out. So we're going to fully oxidize glucose in this final step, or in this third step, the citric acid cycle. During the citric acid cycle, we're going to make a net of 6 NADHs, 2 FADH2s, this is another electron carrier, and 2 molecules of ATP, okay? And that these electron carriers, again, are going to go, just like the rest of these electron carriers, are going to end up in the electron transport chain to make ATP by oxidative phosphorylation. But here, ATP in the citric acid cycle and glycolysis are made by substrate level phosphorylation. Okay, so what are we going to focus on today? How acetyl-CoA goes into the citric acid cycle and how carbon dioxide is released, how these electron carriers are made, and how ATP is made, okay? And this again occurs in the mitochondria in eukaryotes and in the cytosol in bacteria and archaea, okay? And then here's the CO2 for every 1 turn that comes out. And then here's the NADH and the ATP and the FADH and the NADH that's made as this cycle turns around. Okay, so we're going to walk through all of this. Okay, so we start off with our oxoalloacetate, which is a 4-carbon molecule, and it's going to combine with our acetyl-CoA. And this is going to make citrate, okay, and the carbons are going to come at the end of this molecule. Okay, it's going to isomerize to isocitrate. And then 1 molecule of carbon dioxide is released, and 1 NADH molecule is produced. And we end up with 5-carbon alpha ketoglutarate. Okay, so here's a carbon coming off. Then another CO2 molecule is released, and we end up with a 4-carbon succinyl-CoA, and another NADH is produced. Then ATP is produced, or GTP, by substrate level phosphorylation, and we end up with succinate. Then we get 1, as this is oxidized further, we get 1 FADH produced by reduction, and we get fumarate. And then 1 more turn, and we get NADH. Okay, so here's where for every acetyl-CoA, we get 1, 2, 3 NADHs, 1 FADH2, and 1 ATP or GTP. Okay, and you can see now this is the next turn, right? And so now that first acetyl-CoA ends up in the middle of the molecule. Okay, so none of the acetyl-CoA the original acetyl-CoA has been released until the third turn. The third turn of the cycle, when finally the acetyl-CoA is at the end of the molecule, and here our CO2 is released. So it takes 3 turns of the cycle to get acetyl-CoA to come out. So again, we're talking about the oxidation of glucose, but other metabolites can enter in the cycle here, particularly proteins. And so here we can have proteins, amino acids enter in and be converted into acetyl-CoA to enter into the cycle. Okay, so all food can be metabolized in some way, and maybe it'll enter in before glycolysis if it's a sugar, or maybe proteins will enter in as pyruvate or acetyl-CoA or directly into the citric acid cycle. And same for fats, right? So fats can enter in as acetyl-CoA. And parts of these can also be used up in other anabolic pathways. So here's how everything fits together, where all the food goes that we eat. So how is the citric acid cycle regulated? It has 3 points of regulation. So the number 3 reaction, 4, and number 1 are regulated. So this step number 1, in which acetyl-CoA combines with oxaloacetate to make citrate, is regulated by ATP. So when the ATP levels are high, that enzyme is shut off. And these steps are regulated by phenolic inhibition by both NADH and ATP. So when these are high, these reactions are shut off. So we've now talked about what happens to all the carbon that comes out, right? So we had glucose, 2 NADHs, 2 ATPs, 2 pyruvate, 2 NADHs, 2 CO2, as it's converted to acetyl-CoA, and then it goes through the citric acid cycle to make 6 FADHs, 2 FADHs, 4 CO2, so here's our 1, 2, 3, 4, 5, 6, 4 plus 2 is 6 carbons come out, and 2 ATP. So we've only made a little bit of ATP, really a small fraction, 4 molecules of ATP, but we're going to make a lot more, okay? So we have these NADHs, 10 of them so far we've made, and 2 FADHs, okay? And these are going to go to the electron transfer chain to make more energy. And we're going to use the electron transfer chain and oxidative phosphorylation, and we'll talk about these in the next class. But that's all for today, and I look forward to seeing everyone in class. Thanks, bye.